line will be I'll talk about joint work with Jake Mardikin and Max Kuhn on early prediction of fibromyalgia using electronic health records. And then I'll also give a brief overview of spectrum of predictive models that we applied to neuropathic pain in an observational data and joint application with Cal Johnson and Max again. And then I'll finish with a recent application that we've been working on with Optum colleagues at Pfizer, a wide and deep learning application to identify the cardiovascular events from the claims data, specifically from the emergency room claims data. Now, I'm not going to go into the technical details. I'm just going to go through and erase some issues. And I teach a class at Columbia, and one of the first part of this class that I actually try to emphasize a lot is the overall analytical process. And so for the statisticians, this is nothing new, but it is basically important to actually just touch base and mention this for a brief moment, and then to understand this very clearly. What happened is that for all the projects that I'm talking about, there is always some group that we actually work with, either the marketing groups or the statistics group or outcome and economics group or medical group. So we actually get together and formalize the question and objective of what we are trying to do. That is very key because that step actually clarify the questions and then sets the goal and objective in a sense that we know exactly what we want to do and achieve. Next step actually is then to look into the database and see what we have available and whether that's going to be enough to address the objectives that we set forward. So the database comes in, then the pre-processing, cleaning the data, and putting the data in a way that actually is ready for the analytics or analysis is basically the most important part of the putting everything together to run the models. The easy part actually in some sense is to just let once you decided what you're going to do and once the data are ready to run the models. That might take some time depending on the algorithm, but then that's basically a much easier part of it than putting the data together and cleaning and pre-processing whatnot. Then the process then keeps going because once you have the results whatnot, you need to actually communicate those. So how are we going to communicate? And I'll actually try to go over some examples and that communication, making sure that people can understand that, because there are different backgrounds of your customers, and based on that, you need to make sure that these are understandable, the results are transferable, and basically people can get something out of it. And last part is basically making sure that this analytical process at the end, you get something out of it. So you evaluate, from the interactions with the customers and see, is there something missing? Can you update this? And can you basically do a better process next time? And then just, this process keeps going and going. But just basically this overall analytical process is key, and we apply this and try to apply this to every project that I'm going to talk about. So this publication that we did on fibromyalgia was about 2015, when the big data and the electronic medical records and machine learning were huge words. Everybody was talking about big data. Everybody was mentioning big data. And actually, it was so big that Forbes magazine had an interview with one of our executives from Pfizer. And the idea that, that the work that we did actually was recorded there, and it was mentioned in the Forbes in a very positive way that, we're using the electronic medical records based on big data, and we're using the machine learning algorithms to help basically diagnose a disease that's very relevant. It's a chronic disease that's relevant to individuals that we deal with. So from that perspective, fibromyalgia basically is a disease that is a chronic disease that actually is very costly, and it's not very easy to diagnose. And it it takes on average about 5 years to diagnose fibromyalgia patients, and then it actually has a lot of comorbidities attached to it. And And idea was that if you can, use the electronic, some of the data mining methodology using electronic record models from, from an insurance or payer's perspective, can we diagnose these patients early enough so that whatever treatment out there, these individuals can utilize and so that they can actually get feel better. The goal actually is, again, just to develop a predictive model to diagnose fibromyalgia patients. And we use the data from electronic medical records from a Humedica database. This database is across the United States and has a bunch of healthcare systems that's attached to it. They collect data, and then these data then aggregated and de-identified as part of the electronic medical records, and that actually includes demographics, it includes the healthcare utilization, it includes lab data, and a bunch of other things that you can actually think about it. So this, the inpatient visit, emergency room visits, the labs, the prescriptions, all those data are available from that for each of the individuals that we have. We had a protocol, and in the protocol we specified some inclusion and exclusion criteria, and afterwards we ended up about 600,000 patients that actually meet our criteria. And what we did actually is we said, we did some benchmark models, such as logistic regression or some others, easy ones that we can actually work with, but we also wanted to see something like random forest, how that could actually process and work with this kind of data set. We split the data as a training and test. So we put about a quarter of the data as a test or validation, and then just to get a so that we can develop the model on the training data set, but then test it on this untouched validation part and get an honest estimates of how these models actually work in terms of accuracy and classification purposes that we are dealing with. Just a little background on the random forest. It's basically an ensemble method developed by Bryman, and it's very similar. Well, it actually uses the classification trees on the bootstraps that we've generated. In this case, we use actually 1,500 bootstraps of the sample that we have, or the data sample that we have. And then the random forest actually works in a way that at each of the bootstrap, it constructs a full classification tree, but actually utilizes a randomly selected number of variables or predictors to do the classification. Now, one other issue is the class imbalance. When we actually had this data set available to us, we noticed that less than 1% of the people were diagnosed as having fibromyalgia in our data set. So that actually creates an issue, because now the model not only needs to predict does a good prediction in terms of accuracy, but also needs to deal with the class imbalance that it's presented in itself. So talking to Max and the others in the process, we decided that we will actually employ some of the methods known for us to handle the class imbalance. Some of them are the downsampling. So what that means is that you look at the basic cases, the fibromyalgia patients in this case, and then you randomly select the same number of or similar number of non-fibromyalgia patients, and then do your model development on that downsampling, just basically concentrate on that set, and then you can do upsampling, or you can do a hybrid approach, whatnot. And then we've tried all those, and we decided that random forest actually has this nice capability of doing internal downsampling, and just basically using a stratification in the code to actually let us utilize the downsampling approach very simply, and that's what we've done, and we prefer that because it actually showed that of all the training data sets that on the training data set that we did this, the downsampling actually was given one of the best accuracies for us. we use the area under the curve, by the way, to measure this. And then just to get more detail, we use cross-validation, and this was a tenfold, but repeated 5 times. So, this is just, you can imagine that this becomes a huge, intensive computing process that's going to go through, and the number of random variables that were selected optimized at 13, and we basically use that in our implementation of the random forest. So just with a little background, we did the analysis, and then we look at the variable importance, and we noticed that after 10 predictors, the importance actually were decreasing remarkably. So we said, all right, why don't we just concentrate on the top 10 predictors and see if that actually does a hit on the compared to every variable that actually is available to us. And we noticed that we're not getting too bad of a hit. So we then incorporated only the top 10 predictors, ran a random forest on that, and then the training data set actually shows that we are achieving about 82% area under the curve with downsampling, and then with a very reasonable sensitivity and specificity improvement that we've seen. So with that, we said, all right, that's the training. Of course, hopefully there's not much of a overfit issue that we're going to be dealing with. So let's just make sure that this works on the test data. Remember the quarter of the data we set aside, and in there we actually found that the model actually still performs very good, 81% in the area under the curve, and that's basically we said, okay, this is the model that we're going to go with. And by the way, compared to the benchmark logistic regression, this provided about 10%, 0.6, but about 0.65 or so about 15% improvement on the test data on the accuracy wise. So this is a preferred way to go. Random forest, again, doesn't really require too much pre-processing, and then you don't have to worry about too much of the multi-collinearity issue. So there are a bunch of benefits that we incorporated and in the utilization of the random forest. However, communication of these models is not easy. So it's a complex model. It has a large footprint, meaning that there are zillions of thousands of if and then clauses that you can think about it. It's not easily interpretable because you're assembling these trees across 1,500 bootstraps that we've done, full trees. And what became an issue is how can we make the practical prediction, and what is it that we can actually communicate this? And then we said, all right, you know what, why don't we actually do a web portal with a nice interface? And that's actually one of the reasons that we actually come back and talk about the top 10 predictors, because this is basically a portal that we outsourced to a company. We provided the random forest object from the R that we ran, and then in a way that you can actually see that you can actually provide the 10 variables, and with sliding scale of whatever we have available to us, each of these variables, and then on this side you can populate these parameters, and if you say compute, it actually gives you the predicted probability of having a diagnosis of fibromyalgia or not. So in this case, a 78% chance that this patient with these characteristics will have a fibromyalgia. So, and if that exceeds the general accepted cutoff of 50%, you say, all right, this is a high-risk patient, so 78 is high enough, and that's what we're going to utilize. Now, the other option was, since it's a random forest, you can do individual prediction, but if someone actually provides you 10,000 patients in an Excel spreadsheet, obviously you can still do the prediction. It'll take maybe a bit longer, and we provided that as an option as well. So there's a population tab where you can enter a bunch of spreadsheets of individuals with these 10 characteristics or predictors as an input, and it'll actually then provide you a predicted probability of fibromyalgia for all those patients. And we actually have utilized this with some healthcare. So I just want to see that, and this is the population work. Now, this is great because what happens is that now individuals can actually go through and use this. But in the paper then, we wanted to communicate some of these, and then we said, all right, so we can provide the details of the portal, but let's make sure that the manuscript can give some idea too, and that actually the C50 rules actually come out. So we provided in the manuscript some rules that are meaningful and has a nice list actually instead of the area under the curve. We look at the list, and we found some simple rules that you can actually use as basically some guidance, as prediction guidance for you in the manuscript as well. So in the second part, we use basically a study from a 6-week perspective, non-international study of patients on neuropathic pain with pregabalin in Germany. This was about 15,000 patients, and again, we had some kind of imbalance issue there. Our interest was how can we predict the patients achieving at least a 50% pain reduction by week 6 after treatment initiation with some drug called pregabalin, and we actually use a spectrum of machine learning algorithms and some basically, linear discriminant analysis, kind of simple stuff, naive Bayes and whatnot. But and then again, we just utilize the tenfold repeated 5 times in this implementation. And then each method has their own tuning parameters. So I'm not going to go through that. And so one of the interesting things is that what are some of these baseline characteristics and clinical characteristics that we have. We noticed that we can actually put something post-baseline into our model. We decided that we're going to use the treatment at week 1 and week 3, and then just see if that's going to actually make an improvement on basically on the data. And then just to go through this, just briefly concentrate on the bottom shell, the C here. These are the performance that we are observing from these about 11 models that we put together. And we noticed that most of the models are actually doing fairly well, and the conditional trees actually are basically giving us somewhat small results and smaller values in terms of accuracy. But they were all about the same level of accuracy, 89% highest with the LDA, and random forest and boosting is about 88. And this is, again, just another application that I wanted to just to give you a sense of what we do. And then the last topic that what I want to talk about is we wanted to use deep learning on a classification model to predict who will have a cardiology-related emergency department visit or not. So there are problems because we want to examine the prevalence, and we also wanted to look at some label noise impacts on the model performance on the data set. The data came from Optum claims data, and it actually utilized clinical, that includes some demographics, whatnot in there, as well as the natural language processing and text mining has been done actually from the unstructured clinical notes, and then they were extracted and put into this database as part of the predictors. And so what we have done actually is that we looked at different volumes of emergency department visit data sets. We basically wanted to see if you change the number of cases and controls and then add some label noises, how the deep learning actually may behave and give us performance depending on these different parameters that we are interested in. We applied a few models there, and one of them actually is called the wide and deep learning. So the wide part actually is coming from the just regular regularized regression. In this case, it's a lasso that we've implemented. And then it's a joint model of something like lasso versus a deep model, a neural network, which we had put actually 2 layers in there. And we tried some other methodology out there too. But we split the data at training and test, and then validation. And I'm just going to talk on the deep model perspective. We put 2 hidden layers, and then from there, in order to talk about the all fitting, because we didn't do cross validation, we use a learning rate we implemented a drop rate and then also did an early stopping, and here are basically some results that I'm showing you. So let's just concentrate on the area under the curve. With different, when you actually the prevalence is about 50%, meaning that the case and controls are very similar. The area under the curve was about 79%, and when we actually did a class imbalance, we forced a class imbalance into the data. We noticed that we can still employ some of those methodologies I mentioned earlier in terms of downsampling, upsampling, but in this case, upsampling actually basically was the method to go, and this actually improved our lot. Actually kept the results around 80% area under the curve level. We varied the oversampling, but also incorporated some label noise into the picture. So different label noises, 10%, 20%, and 30%, where we actually just take those cases and randomly made them control, and then we wanted to see how the deep learning actually affecting or working in these scenarios, and it seemed like it was actually performing fairly reasonable. So I just wanted to talk a few minutes about some of this work. The fibromyalgia work is the first reference, and then the spectrum of the methods is the second reference, and then we utilize heavily on Max's book on applied predictive modeling as well.
Thank you much, Sir Birol. Yes, thank you so much. That was a tremendous amount of material in a short amount of time, so that was a great landscape picture of the machine learning work that you've been doing, and I hope it gives our participants a great idea of the great work that's being done in pharma and machine learning. In the interest of time, let's go right to Junshree's slides. So I'll bring those up, and I will pass the ball to him. And I'll remind folks that to ask questions, please use the chat, and we'll have time for questions for both presentations at the end. So I pass the ball to Junshree. You're up.
Okay, thank you, Dan. This is talking about using deep learning to predict a model molecular activity with its structure. And indeed, the underlying question is more like deep learning. Is deep learning an evolutionary or revolutionary solution to this particular problem? This work is in collaboration with my colleagues, Yuting Xu, Andy Liao, Bob Sheraton, and Vladimir Svetnik. Here. Right. So in this talk, I will first briefly talk about the background of this whole effort, this particular project. And then after that, I will focus on two particular topics regarding the application of deep learning to this particular area called QSAR. Indeed, you see QSAR is a research area to study the relationship between molecular structure and its chemical or biological activities. I will use next slide to make this sentence make more sense to most of you. And then the last item is about summary discussion of this effort. Yeah, this is a general kind of a pipeline for small molecule in a pharmaceutical company. Their kind of a development, research and development pipeline. And what we are focusing here is this particular area called lead identification and lead molecular optimization. Especially in the lead molecular optimization area. It has covered a lot of kind of areas, like, for example, how can I make this compound more potent regarding the target? For example, the target is a particular neuroscience target or cardiovascular target. How can I make my compound more potent to that target? And also, how can I make the compound more like a drug? When this molecular is a drug, means this should meet some ADAM property. ADAM means this kind of full air kind of a property regarding the drug work interaction with the body, and also how can I make the compound less have a less safety issue. So there are a lot of this kind of question you have to answer to make sure this molecule can be used as a drug. All those things is kind of a measured most of the time in the past in the lab, and what they call the result of all those measurements called chemical biological activities. And basically the QSR is the area of using the structure to get the molecular activities. The activity is what I was saying a moment ago, all those items. In the past, those was done by lab experiments. Even right now, I think the gold standard is still lab experiment. But of course, in the modern world, what we really want to do is we want to use some computing kind of a predictive model to just input the structure of a compound into a predictive model, and then use the predictive model to predict the molecular activity, just like you did in a lab. And then based on the activity, you can rank all the candidate compounds you have, and then give the the top ranked compound higher priority to do follow-up experiment. And at this moment in this field, this is the area like the correlation between the lab result and the computer kind of predictive model. It's range from a very bad situation where it can be like a 0.3 to almost kind of like a reproducible kind of situation with a 0.9. So totally depends on the particular problem, particular data set. They can be quite different. Like in some cases, the computer can give a result which can reproduce the lab result. In some cases, it's far from the lab result. So that this makes this whole area still a research topic. And in 2012, Merck, like a group of Merck scientists, put all these 15 very diverse Merck QSAR database to a global computer science competition platform called Kaggle. I think a lot of you heard about it. And basically, this is a platform people all around the world to solve the problem and compete to see who is getting the best result. And this is the 15 data set that we are providing. It's very diverse. Some of them is about target potency, some is about atom property, some is about safety, some is about efficacy. And then at the end of 2012, the result, I mean the winner of this competition was a group using deep learning method. George Dahl was a kind of a key member in that group. He was a graduate student in University of Toronto under Dr. Hinton. And our internal method was a random forest. And the difference between random forest and the winning result is about like random forest give correlation between the lab result and the prediction result is 0.65. The deep learning is 0.7. Indeed, we consider that a fairly significant improvement. This is a result by average all the 15 data sets. And that the whole thing was even kind of published kind of partly in this New York Times article. So now let's move on to kind of the fundamental questions like this is the first time the deep neural net, deep learning, produce a result that is better than what we have as a random forest. So the question is like, is this deep learning method representing a revolution, kind of a revolutionary kind of a start of the new age, or it's just evolution kind of improvement as another method which happen to be better than random forest? So let's see what's the winning method in that competition. So this is a winning method. Basically, it's kind of a deep neural net. When we say deep, means that you can see, for people who are not very familiar with neural network, I will just kind of spend a few more sentences to explain it here. So the first layer is, we call that input layer, and that's basically all your features. In this case, a data set can have 8,000 features, very sparse features. Only 5% of them are non-zero, so most of them are zero. So you push this 8,000 features into the input layer, and then each row here is basically we call it a hidden layer. In this case, we have 3 hidden layers. Each hidden layer can have a few thousand hidden nodes, and then this build up the network. The output is kind of a 15 output, which is like 15 tasks. Because we have a 15 task here, so each output can produce a result for each particular task. So this is a winning network. What's unique about it is winning network. It's here. It's traditionally the deep learning was doing things like this. It's just kind of have one layer, which is kind of not deep. So the hidden layer is 1, so this is not deep neural net. And also the output is kind of most of the time just kind of one task. So the input of this whole thing, clearly you can see 2 major innovation in this winner method. One is it makes this like deep neural net, which makes the network deeper with multiple hidden layer. Second is it has this multiple task kind of situation, like the output. I produce the output, all 15 output at the same time, instead of one task for or one network for one task. So the first question we will try to answer is this deep network or multiple hidden layer really help? So this is a plot. You can see the x-axis is the number of units in each hidden layer. The y-axis is basically the performance, the difference between the neural network compared with the random forest. This is the average across all 15 data sets. So for example, this dot means the red line means 2 hidden layers. The black line means 1 hidden layer. The red line is more like the deep network. The black line is more like traditional, just 1 hidden layer network, which is happening in the 80s. So, for example, this data point is I'm using 2 hidden layers, but each hidden layer is only have 12 units. In this case, you can clearly see if you have a wider hidden layer, the 2 hidden layer is better than 1 hidden layer. So what we can see here is indeed this area was the kind of people doing in 80s. People in 80s, they are not having the computation power to have a very wide kind of hidden layer. So they focus on the kind of a narrow hidden layer. In that case, they only focus on 1 hidden layer because kind of 2 hidden layers they frequently find it's like run into this like a local minimum issue. But indeed, in the modern world, because we have the capability to use wider kind of hidden layer, then the advantage of this like deeper network is showing. What about a 3 hidden layer? But if we put 3 hidden layer here, you can see the advantage is not that obviously. So our conclusion here is basically kind of yes, deep help, but with a limit. Normally, it's not go beyond 2 to 3 layers in this particular situation. And also, generally speaking, deep network generally require wider network. Okay, so briefly, kind of a questioning, like what's the general kind of reason why this is working. Two series papers. One is published in 1991, another is published kind of recently. Basically, kind of the first one is what we all know. Deep neural net easily approximates almost every arbitrary complex predictive function. The second one is useful in the sense of basically it's saying the large network, the optimization landscape is like what we are showing here. Almost, you got a lot of local minimum, but all the local minimum are pretty good. And but for the small network, you got a lot of local minimum, but the local minimum can be very different from the global minimum. So that's basically what it's saying. But why it doesn't work? I mean, we think because deep neural net is very good in feature engineering. For example, the image, video, kind of a speech, all those kind of things. Deep neural net right now is dominating because they are good at extract features. But QSAR, these kind of molecular structures, you change a small thing over there, the whole activity can be quite different. So it's not, the feature right now is not redundant. So you cannot do much about this feature engineer over there. Probably that's the reason QSAR is not seeing the dramatic improvement seen by other in the sense, under deep neural net. So the next topic is about why the multitask deep neural net kind of work. So what we are showing here is 15 tasks kind of we are putting in the computation. So the blue box is the range for you just train a single network for this problem, and then what's your output. The red one is using this multitask network. You put all the 15 network data, no, 15 task data into the training set, and then generate all the result at the same time using one single network. So what we can see here is, on average, the kind of multitask network beats the single task network. But if you look at an individual case, for example, in this case, the multitask it gets even worse. But in this case, I call the OX2 case, the multitask is highly kind of is much better than the single task cases. So why this kind of thing happened? And so what we are doing is we basically kind of focus on this OX2 situation. For OX task, we are not combining with all the other 14 tasks, make a 15 task network. We just combine the OX2 with individual tasks to make a 14 two-task network so that we can see which task really contribute to the overall improvement. So this is the result. In this result, what you can see is that this is a 14 task individually combined with OX2. And the blue line is the range of we just train OX2 by itself because there is variation. So the result is within these two blue lines. And the red lines are all the 15 you put together to train. What surprised us is it seems like it seems like there is one task. This OX1 just kind of have all the advantage. Like combine this one task is equivalent to like comparing all the 15 tasks. So in this case, you can clearly see the advantages only come from this particular case. So why? Then we go back, look at our data. We found this OX1 and is developed in a period like before the OX2. And the OX2 training set is indeed have about 2,000 kind of compounds, which is existing in OX2 test set and also OX1 training set. And so, and also what more is like the activity between the OX2 and the OX1 is correlated. And so this is kind of the reason it helps. And also the next question is, yes, we know if we have similar molecular structure and positive correlated situation, it helps. What about the uncorrelated situation in this case? What about the negative correlated situation in this case? So then we did the similar thing before. We found if we have data set which is kind of similar with our kind of have in the structure, but correlated, positive correlated, or uncorrelated, kind of negatively correlated. In both cases, they are helping the problem. But if it's uncorrelated, it makes the problem worse, even worse than you just use that data set alone to train the whole thing. So this is the kind of the result of this finding. Basically, kind of we propose in the future when we have a primary training set and solving a primary testing set, we can introduce a system data set. This system data set has a kind of a compound which are having similar compound structure with the test set, but because kind of we know the activity is somewhat kind of related to this, either positive or negative related to this primary kind of problem. And in this case, we know the result will kind of improve just trained by the kind of a primary case alone. So indeed, what we got here is we introduced this system training set, which is a way to incorporate domain knowledge, because the structure-wise, we do know which data set has a compound structure which is close to our training test set. And also because we know the nature of experiment, so we know the activity, whether they are related or not. For example, if the primary problem is about a neuroscience target, and then a system set can be a PGP kind of activity, because that's related activity, so that we can select a system set, incorporate our domain expert knowledge to assist them to boost the performance of primary site. And so this is kind of as a summary of this whole thing. We think at this moment the deep neural net for the QSAR, in the sense of lab reproducibility, we think the deep neural net at its current form is still an evolutionary solution for QSAR. There are kind of two areas we potentially can transform this evolutionary kind of change to revolutionary change. One is incorporate domain knowledge. What we found kind of like a moment ago, the assistant data set is one of the ways we think can help to incorporate domain knowledge. But another limitation we found that a better feature extraction for QSAR problem, that is still something we're trying to explore. At this moment, we tried things like graph, convoluted neural net, all those kind of things, and then the feature was still not good enough to really transform this field in the sense of other like imaging pattern recognition kind of field. So that's it. And this presentation was covered by two papers. I was not listing here. Yeah, that's it. Dan.
Okay. Thank you, Junshui. And again, we tried to set this up so that we had a presentation that was sort of wide, lots of different applications, and more in the clinical area. And Junshui gave a great representation of some of the things that go on in the preclinical area, and a little more depth in terms of how it, how it actually works, and some recommendations in terms of using deep neural nets. So now I'd like to turn it over to Max for his comments and for some discussion.
Thanks. No slides here. I'll just talk about a few things from both presentations and in general, and then we can start with questions. So some thoughts. If you're new to this field of machine learning and predictive modeling, and especially if you're not more on the the science side of pharma, for example, it could be kind of daunting to look at this stuff. And it really takes a change of really figuring out what works and what you're able to do with machine learning models, and we would not typically do with classical statistical models. So for example, these machine learning models are very, very, very good at prediction, but they kind of lack in a lot of other areas. So you probably wouldn't use a lot of these models to determine what is really important in your data. You'd probably be better served by more ordinary conventional statistical methods about ANOVAs and visualizations and things like that. they can be sort of like stunningly bad sometimes at deriving very detailed descriptions of what is driving things in the data. Now, you can do things in a lot of these models like variable importance scores and some other methods that will give you sort of in aggregate what is driving the model. But you're not going to get things like you would from, let's say, logistic regression, where you're going to get things like, increases in, odds of the event per unit of the predictor and that kind of thing. So they're really good for what they're meant for, which is typically prediction. One other thing that is really common in machine learning that is not necessarily common otherwise is to think about the focus on empirical validation. So both speakers really focused on looking at measures of performance, usually related to accuracy, but some sort of predictive performance of their models. And typically what we do there is we use both a test set, which is data that we set aside until the very end of the modeling process to sort of independently characterize how well the models are doing. And another method is to use resampling, which is like the bootstrap or cross-validation. And those are methods during the process of building your models you would use to get good estimates of predictive quality. And so the thing to emphasize there is, number one, those are really, really critical for these types of models, mostly because we're doing things that we're not usually validating or characterizing the quality of the model based on statistical characteristics. So when we think about doing like a linear regression, we think a lot about, what's the distribution of the residuals, what are the distributions sometimes of our predictors, and things like that. It's not that those don't necessarily matter to these models, it's just that we've so focused on empirical validation of prediction that we don't usually worry about it. And so one of the cool things, I think at least, about doing machine learning is you can basically try a lot of other things in your model that, perhaps you cannot analytically derive properties of. So if you put in principal component values instead of the original predictors in your model and then fit, a random forest model, there's really no way of making any assessments, even if you knew the distributions of your original predictors, as to what the statistical qualities are of the model. And so it's just really impossible to do that. Leo Bryman, the guy who invented random forest and was a big part of CART and all those models, he had this phrase of there's no Bayesians in foxholes, meaning that at least at the time he felt that we couldn't really do very sophisticated statistical things in machine learning because they just couldn't handle the functional forms that we need in machine learning. I don't know that that's true anymore, but I think the theme of it's there is we can't really rely on statistical principles when developing these models, or at least they don't matter as much as predictive performance. So one aspect of that, though, is, back when I used to work in pharma, it was very common for me to see people develop logistic regression models, for example, where they spent a lot of really diligent time crafting what features were in there or predictors were in their model based on what was, statistically significant. They're looking at a lot of likelihood ratio tests and things like that. And so they're more worried about the statistical characteristics of their models, but not really the predictive characteristics. So I would see all this work, and then you would ask somebody, well, what's the accuracy of your model? And it might be like 60%. And so, a lot of times in traditional statistics, we can really be well served by borrowing a little bit from the machine learning area and think about empirical validation. There's no point of having a well-crafted logistic regression that's 60% accurate, because any inferences you make from those models, are likely not to have much fidelity to the original data and may not be generalizable. So that, this focus on empirical validation is something that would be a good idea to pursue a little bit in more traditional modeling. One other aspect of this is acceptance of these models. So a lot of times, especially in medical-related applications, what I see is a lot of people not accepting models because for many of these, you can't show them what the slopes and intercepts are. In fact, there might be, orders of magnitude more slopes and intercepts in your models or parameters than there are data points. And that sounds really awful, but a lot of times it's really made palatable by the use of regularization and some other methods. So that's okay to have that, but it's very, very hard to describe in detail what these models do for prediction. And that is a drawback to them. And I don't know that we have a lot of really good methodologies for solving that quite yet. And it's definitely, I've seen doctors say, well, how can I trust this model if I don't know what the effect of, age or blood pressure is in the model? We can show them, again, aggregate things saying, the age or blood pressures were very high variable importance scores and things like that, but it's not really what they're looking for. And, they can sometimes be convinced by empirical validation, really well thought out methodology in terms of how to validate the performance of your model. But again, you can't like on a piece of paper write down what the equation is for the model, and that's a drawback for a lot of people. Now, Brule in his work, which I'm more familiar with, we were able to do some things with the random forest model. Since there was a big class imbalance, it was very easy to come up with a rule that said if you fit into this rule based on your predictors, these types of patients, we knew that you would be predicted as not responding based on that, because there were so many, the class imbalance basically funneled almost everybody into the one terminal node of the tree. So there are some occasions with certain models where you can do things that would help people understand why it's predicting samples the way it is, because they might be drawn to one particular part of the tree, or they might be, if you have one really, really strong predictor, that can be a big help. There's also some other work. There's this methodology called LIME, which is like, for example, you can use in an R package, which does a really good job once you have a model and you make a prediction. It can tell you, or at least give you a better sense of why the model predicted that sample the way it did. It's not perfect, but especially for like a graphical representation of that, LIME does a really good job. That's L-I-M-E, like the fruit. Some other things, especially regarding deep learning. So deep learning is in a really, really good stage right now. Google released a couple of years ago this computing language called Tensorflow. And based on that, people have written a lot of packages that make the fitting of very large neural networks and deep learning models very fast and easy to do. And so that's a really great tool to have. The good thing, so there's good and bad about that. So the good thing there is when you build these deep learning models, they work really, really well in places where you have a lot of data, especially a lot of unlabeled data. So you can imagine in the QSAR example, we have millions of molecules in every one of these companies that may not have the outcome estimated that we're interested in because the lab test hasn't been run. But we know all the predictors because they're almost always deterministically derived from the molecule structural. And so what QSAR models could benefit from is building up what they call auto-encoders, which are the early parts of the deep learning network, that basically they don't learn the outcome, they learn the relationships between the predictors, which in this case would be the QSAR features. And so one really cool thing that you can do is you can get really, really good representations of your molecules based on data that aren't being used to fit the actual model. So you might, again, have millions of molecules where you have their predictor values but not their outcome. And deep learning models are really good at building up those parts of the network using all the data instead of just the data that you have the outcome for. And that's really powerful. Another good aspect of that is, and this is really true in image and text applications of deep learning models, is a lot of people build up really standardized sets of early layers of the network that everybody can then use. So if you think about Google and what they do to do image classification, they have just astounding amounts of data and build up auto-encoders or models that basically model the structure of the predictors. And those are basically locked in place. And so once they're well validated, everybody can use those as an early part of their deep learning model without having to refit them, potentially on much, much smaller data sets. So, the good and bad of deep learning is they are really performant. They tend to be reusable in the way I just mentioned. They can leverage data that you don't have the outcome values on. But on the downside, they tend to be, excuse me, my dog's rolling around. They tend to be pretty high maintenance in terms of model training. To fit those models individually, they're relatively fast compared to other things, but they usually require a lot more care in terms of the predictors that go into them, how those models are optimized in terms of their hyperparameters and things like that. The comment in the second presentation about needing better features, and basically, what was said was we need better ways to extract the information out of the QSAR features. Now, QSAR is one of the places where I'd say the number and the breadth of potential predictors that we have is astoundingly high. It's really good. There are millions of potential predictors for every molecule, and we can compute them very quickly. Having worked in this area before, especially for deep learning models, what I can say is when I've been able to develop good models, you have to spend a lot of time on the predictors and figure out what should be there in what format. So basically, there are a lot of pathological distributions in these descriptors. there's a lot of counts of particular types of molecules or substructures where 99% of the counts are 0, and then you might have smaller numbers of counts with one representation of that substructure, or 2 or 3 or 4. And so there's a lot of predictors like that. They also tend to have very, very high between predictor correlations. So collinearity is like the kryptonite of neural networks. They tend not to do well at all when you have highly correlated predictors. And so feature extraction methods like principal component analysis or independent component analysis can have a really good effect on those models. Now, the reason I bring that up is just an example of something where you have deep learning, which is a really expressive and highly performant model, but you need to spend a lot of time figuring out how things should go into that model, whereas something like random forest or some other models really wouldn't care about that. So I think deep learning models can always give you the best possible prediction if you work on them long enough, whereas something like random forest and boosting and some other methods will get you within maybe 5% of what deep learning will get you, but maybe within a few, with much, much less effort. So that's, to me, the plus and minus of the deep learning models. So, I would say if you're not used to, so in summary, if you're not used to machine learning, both these presentations are a good start on examples and what, we consider to be important and maybe what's not important. There were no p-values really in these presentations and things like that. The great thing about machine learning is there are tons and tons of resources out there, both to read about and to start using all these methodologies. There are commercial software to do these things, but whether it's in Python or R or in Tensorflow, there are a variety of tools that you can use to get started. So it's the kind of thing where if you want to get involved in these types of this type of modeling, there's no barrier to entry, which there would be in some other areas. So that pretty much summarizes my thoughts. I think we're going to go to questions, and I have a question for both the speakers. So does that sound good for me to start on that?
Yep.
Okay. So Barul, I had trouble remembering when I was working with you on this stuff. Was there any pushback from the clinical people about using this big tree ensemble to making predictions?
Yeah. Thank you. We actually basically sent this format of what do you call the web page portal, and it's called populationdetect.com. And then we asked the insurance and payer companies to actually upload their data sets. So I think there were a couple examples, like University of Pittsburgh Medical Center. They actually put some patients in there in thousands, and then they, with those 10 characteristics that we recommended, they upload that into the web portal, and then concentrated on the top, let's say, 20%, those that have a predicted probability of fibromyalgia, about 80% or more, and then they were actually trying to access those individuals and see if they should actually see a clinician, a physician, to see if actually fibromyalgia really could be a concern, and so that they can actually get a therapy right away.
But nobody pushed you back to say why don't you just use logistic regression, that kind of thing? They
did. Well, actually, they were actually asking other things. They said, well, why don't you use the BMI? Why don't you use this variable or that variable? And that actually, again, depends on the availability of what you have with respect to the electronic medical records. We tried to actually incorporate some of those already. Like BMI has 80% missing value incorporated in it, and then they ask about some other models that may be available, and we did actually have a logistic regression as a benchmark because we wanted to see, if the random forest, for example, how above and beyond is going to perform against the logistic regression. If the performances are similar, why not go with something that's very, interpretable like logistic regression? And yeah, we had those kind of questions as well. More of a question like, why don't you include this variable or that variable? And then the idea was that, well, we really didn't have access to it, and in most cases it was missing most of those values, so that's why they were not incorporated. Those are the kind of questions that we were faced.
Okay, thanks. And so my question for the second speaker would be, can you talk about how, given the workflows in QSTAR that you guys already have for building your standard QSTAR models, how much, if any, did the deep learning sort of infrastructure impacts like model updating or the day-to-day work in building those models.
Right. Yes, we know deep learning is computational expensive, and that kind of indeed this is kind of the major effort. When we got the competition result, we can have a code, and how can we make that be practical in our daily operation? That's a major kind of internal effort here. What we did is we went through all the kind of potential kind of variables kind of setting, because as you implied, deep neural net required a careful tuning of the parameter to get a meaningful result. And we end up found a set of kind of a setting that is kind of like universally universally pretty good for QSAR problem. So then that makes this kind of this deep neural net kind of practical to be used in this daily operation, because we can't tune parameter every day. So, and also we cannot tune parameter for every task we put over there. So once we find this kind of a reasonable kind of universal kind of a parameter setting, we just fix our deep neural like that, and then our criterion is all the tasks should be finished within 24 hours. And we achieved that with kind of a simple kind of GPU card. So at this moment, I think deep neural net is kind of used as one of the tools in our kind of machinery, kind of this QSAR machinery. And nobody will feel like it takes too long, or nobody feel like it's too hard to use, because they constantly just use one set of parameter. And the result is kind of, although in the competition across 15 data sets, it generates on average the best result, but in individual task, you still see sometimes the random forest get a better result, sometimes deep neural net get a better result. So it's one of the tasks and one of the practical tasks inside Merck. Yeah, Max, answer your question?
Yes, absolutely.
Yeah.
Should we open it up for audience questions?
Yeah, so there were a couple of questions that came in. One was about missing values, and the other about using RNN or CNNS. So either one of those, whichever one you want to queue up, Max.
Yeah, so missing values is another place where I think we tend to depart from traditional statistics. So when you think about, one of the first things you would think about doing is imputing. Now, when we impute missing values, we usually do multiple imputation in more traditional statistics. And the main reason for that is to make sure that we get the variation due to imputation and everything propagated into the test statistic distribution. So really, again, there we're focused on doing multiple imputation to make sure that our statistical distributions at the end are at least known and quantified. On the machine learning side, it's a little bit different. We tend to do single imputation, and mostly we would, or at least what I would advise people to do, is to include imputation inside the resampling scheme. So it's really, really important to resample all the steps of your modeling process, and that includes imputation and feature engineering and any estimation procedures you do. So, in effect, I think in machine learning, we tend to do single imputation, but we tend to do it many, many times just to make sure we capture that variation in our resampling statistics. So what you would do is you would take, our training set is the data set that we use to build our models. We would build our imputation models on that, and then when we go to impute data used for modeling or any new data in, let's say, the test set, we would use the imputation strategy that was estimated from the training set. So we never re-estimate anything really in the test set. It's really important to make sure you avoid this concept of what we call information leakage, which, basically you can think of defining everything you're going to do in the model inside of a box. And that box is everything that gets resampled during bootstrapping or cross-validation. And that way we make sure that the good or bad aspects of imputation are factored into the resampling statistics as we do it. So we can do the same thing in terms of dealing with missing values. And I should also say there are some models, some tree-based models, and some other models that are very, very tolerant to missing values and don't need imputation. So there are cases where you don't even need to do imputation when you have missing data. But for the most part, when you do, you just want to make sure you include that inside of resampling. So even though you're doing a single imputation, you can quantify its benefit or detriment to the model.
Great. And do you want to have, we have just a few minutes left, but any comments on these different types of neural nets? I think the question really is more towards Birol, like what type of neural nets did you use, and as well as maybe to Junshui in terms of RNN or CNNS or other types?
Yeah. Birol, you start.
I'm on mute, sorry. Can you hear me now?
Yes.
Yes.
So yeah, in our case, we actually are using RNN. That's on works right now, and we're actually trying to see how that actually helps when we incorporate some repeated measures or some longitudinal data that actually is in there. And then, but with the deep neural network that we have utilized, we just use regular with 2 layers CNNS, and that actually was we were trying to incorporate in our models. But we have something called retain actually that we're incorporating as well in RNN.
Yeah.
Thanks.
Right. Yeah, we are using this kind of feed-forward fully connected network, and which is the simplest kind of a network. Indeed, kind of this network is how to say that. I mean, it's not that good. It's not that good. And in the sense of kind of other area, we are trying to use this graph CNN to extract feature, and that may see some hope, but not that great. And also, we tried this kind of Auto-Encoder, and I think it's not kind of wonderful. I mean, so far, I think deep neural net have a lot of hype, but they are very good at image, video, voice, text. But for QSAR, we just feel like it may be just one of the methods at this moment. Yeah.
Great. Thank you. So I think we are at the end of our time, so I want to say some thank yous. First of all, to our presenters and our moderator. I think you've done a great job of enlightening the folks out there in terms of what's possible with machine learning and some of the examples that have provided a lot of value to the work you're doing. I'd also like to thank our attendees, especially those who asked questions. Nick, NISS, and Merck for sponsoring the session. And then a few special thanks, Christy Twang-Stein for helping organize, and Kara Hackman for helping with the technical parts. Lastly, I'd like to say that we always have a short survey at the end of this. That helps us in terms of gauging the time. For instance, we added a little time to this one because of the request from the survey. Also, we're always looking for topics, and in this particular survey, we're also giving you an opportunity to give us your email address in case you'd like us to send you notices of future meetups. So with that, this is Dan Holder. I'd like to sign off and wish everybody a great day, and thanks for participation.
Thank you.
Thank you. Bye.